everything back in class today. Okay, that doesn't usually happen. Usually it's the multiple choice test and it takes me the weekend to get the lab exams graded. But I have 2 more lab exams and another message exam to grade over the weekend. My ARS class. I don't schedule them at the same time on purpose. It just has to fall that way. Today we're starting the nervous system, which, as you know, we've already been talking about spinal nerves and their correlation with the growth of muscles. We will have a reference to that, a lecture where we talk about spinal nerves and look a little bit more at their organization and their location. So we will have a lab where we will be identifying the nerves. And then on this unit in lab exam, identifying the nerves will be part of it. And like you've had a few questions on the lab exam asking for the function of a muscle. You might have, you will have some questions where I identify a type of nerve and then say identify a muscle or compartment of this nerve innervation. Okay, I feel like that, not a lot. What I want to do today, normally I talk about the spinal cord in lab. With Dr. Lancaster covering the lab, I want to make sure all of us have, if you couldn't cover it, certainly could, but I just want to make sure we have the same content. And so I'm going to try to save some time at the end of lecture today to go over the spinal cord. In lab, I will give you a cross-section of the spinal cord, and I believe it's uploaded, copied over from last semester, cross-section with the diagrams of the neurons on it that I will diagram in lecture. Okay? Your quiz on Monday, then, is from that diagram. And it will be, I've done this in the past, it will be identify the structure of the spinal cord. Is it a motor or a sensory component? Does it contain motor or sensory neurons? Okay? So I want to, because that's our quiz, I want to go over that with you in class. The first thing we're going to look at is the organization. How is the nervous system organized? It's organized in 2 ways, all right, by location and by function. So when we talk about its organization by location, we use the term central nervous system and peripheral nervous system. And there are some diagrams to illustrate this on the lecture notes. The central nervous system refers to that part of the nervous system which is enclosed within a bony compartment. Remember we talked about the dorsal cavity at the beginning of the semester? So central nervous system is going to include spinal cord, and brain. So if we diagram that schematically, you would have the brain and brain stem. Brain stem being those structures, the midbrain, the pons, the medulla, right? Babies can be born with only a brain stem and not a brain and still be alive, because within the brain stem are the centers that control the heart beating and breathing. Right? They may not live for very long, or they may live for several years. But they don't have the cognitive function. And so that's within the brain stem. All of that being within the skull. And then starting at the foramen magnum, we have the spinal cord. So that's central nervous system, and then everything outside of that is peripheral nervous system, and that's what we refer to as nerves. And this involves both the brain and the spinal cord. So we have extending from the brainstem neurons that go to muscle, typically in the head, the tongue, the hypoglossal nerve, the facial nerve, the trigeminal nerve, motor neurons going out to muscles. And then we would have sensory neurons bringing some information back. All right. So these would be identified as cranial nerves. And in our lecture, when we talk about the brainstem, we'll be discussing the 12 cranial nerves and what they do. So these would be nerves traveling into and out of the brain via the foramina that we discussed when we talked about the skull. So we have the foraminal valley, the foramen rotundum for the trigeminal nerve. Jugular foramen is going to contain cranial nerves 9, 10, and 11. Hypoglossal canal had our hypoglossal nerve. Okay, so that's where we see the passageway of cranial nerves. And then we have 31 pairs of spinal nerves. Again, motor neurons whose cell body are in the spinal cord, and sensory neurons whose cell bodies are outside. And so these are going to then be considered spinal nerves. And they would be on both sides of the body, obviously. So the cranial nerves and the spinal nerves comprise our peripheral nervous system. And that's by location. Okay? So brain and spinal cord belong to the central nervous system. Cranial nerves and spinal nerves belong to the peripheral nervous system. Now obviously they're not entirely separate. There's not like there's a big gap, a structural gap between them, because the cranial nerves are going to enter or leave from the central nervous system. And if I'm going to move my tongue, all right, I have a nerve that actually has its origin in the brain that's going to tell the peripheral nerve what to do. So there's this connection. Sensory neurons come in, carry information into the spinal cord, and go to the brain for processing. Okay? That's simplistic. There's actually more than one neuron involved in there. But there's this overlap. So the location is just a physical structure of where we find those components. Then we have an organization plan based on function. And I've mentioned that already. As you can see in the direction I have the nerves traveling, we have neurons carrying information towards the spinal cord and the brain. And we have neurons carrying information in terms of electrical current away from the spinal cord and away from the brain. And so those two pathways are two functional organizational components, are sensory and motor. Now, if we were a single-cell organism, we wouldn't need a nervous system. It would be one cell, something would change temperature or ion concentration in the fluid of our environment and would either move towards it or move away from it, because we would have receptors detecting that on the surface of our single-cell membrane. As we become a multi-organizational cell, multiple cells in our organism, then one cell can't take care of all of that. And so we have receptors, pressure receptors, chemoreceptors, we have pain receptors that are detecting our environment either within the body or outside the body, and that sends a signal to the spinal cord and then to the brain and allows the body a motor response. So when we talk about a sensory and a motor organization, that's not separated by location. The brain has both sensory and motor components. Spinal cord has both sensory and motor, and our nerves can be either sensory or motor. All right? So that kind of organization is across the board. It's rather self-explanatory and simplistic, but I'll give you, on our exam, I'll give you some examples. Eating too much pizza last night, and having stomach pains, All right, those pains would be sensory or motor?
Sensory.
Pretty obvious, sensory, okay? Having diarrhea or throwing up afterwards is the motor component, all right? Now, what I described, was that your skeletal muscle that was working, or was that smooth muscle? Smooth muscle, okay? So that gives us a further separation of somatic body wall, which is our skeletal muscle in our skin, and viscera. So what we've been talking about, the nerves that we've been talking about with the muscle unit that we just finished, had to do with the somatic body wall. All right, so we have somatic stimuli and visceral stimuli. See if I can look at what put this here. Now the terms afferent and efferent, I have them under separated and braided spinal cord. I don't want to use the same colors because they really aren't components of those. But this is so our efferent information is information that is leaving, going away from a central location. All right. So this would be illustrated by the neurons that had their cell bodies, and I'll redraw this in a different color, that have their neurons and are carrying information away from either the spinal cord or away from the brain. So you can see that we have efferent neurons in our cranial nerves and in our spinal nerves. But we also have motor neurons in the central nervous system. Okay? So efferent means carrying information away from. And these would be our motor neurons. Direction of electrical current travel is in that direction. Okay, afferent, spelled with an E, spelled with an A, are going to be sensory neurons. And they're carrying information towards the central nervous system. I'm going to kind of put peripheral nervous system here in a block, because I don't want you thinking that the afferent and efferent are separated by the peripheral nervous system. So we have an afferent division, and hence we can get sensory stimuli from those two regions of the body I was talking about. So we have somatic stimuli. This would be information coming from the body wall. So what can I pick up from my external surfaces? I can tell pain, touch, pressure, temperature, there's another special one called proprioception, which means joint sense. You can put itch in there. There's a few others. Anything major I'm missing here? Okay, that's a special sense, and it requires organs to do so, the viscera, so I'm going to save that for something else. Then we have visceral stimuli. and that refers to body organs. So visceral stimuli, we certainly can have pain. Touch is not, I mean, I don't feel when I swallow something, I don't feel it hitting my stomach wall. All right, I don't have the concept of the food moving through my digestive tract or blood flowing against the walls of my blood vessels. When my heart pumps and closes the valves, I don't feel them close. All right, so touch is not really something that we have associated with the viscera, but we do feel pressure. All right, so you can feel a heaviness, you can feel pressure, chest pressure, pain, you can feel gas, putting pressure on it, and that can be changed to pain. Temperature, you swallow cold water, you lose the sensation, it doesn't feel cold, it hasn't gotten body temperature by the time it reaches your stomach. But again, you don't have that sensation of cold in your stomach. All right. My father-in-law, who recently passed away, had a stomach tube in his place in his abdomen and his stomach, obviously, because he was getting aspiration pneumonia. He wasn't swallowing well. They took him off crackers and bread and things that could crumble, but he was still going in the hospital for pneumonia every 4 or 5 weeks. And so they ended up putting, and we should have asked him, what do you feel? Well, because it was just large syringes, and they'd inject via the plunger the food directly with the liquid directly into his abdomen, into his stomach. But typically when we swallow, we don't feel that sensation. We certainly don't have joints in our organs, all right? So we're not going to feel those kind of sensations. But somatic means from the body wall, visceral from the organs, all right? And then these are going to be our afferent stimuli. And they are going to go via the peripheral nervous system, either via cranial nerves. Your mouth, by the way, is considered part of somatic, because you still have skeletal muscle forming that. And they're going to go via spinal nerves as well, into the central nervous system, all right, spinal cord or brain, into those components. So both of these, either via cranial nerves or spinal nerves, can end up going into our spinal cord or into the brainstem. So that's our sensory or afferent division. Okay. Then we have a motor division, the efferent division, and that can be divided into two areas as well, somatic and visceral. Our somatic is what we just got through using with our skeletal muscles. So somatic is actually going to be our voluntary muscle. And then our smooth and cardiac is going to be controlled by the autonomic nervous system. And that would be smooth muscle, which is what we have in our blood vessels and what we have in our irises of our eye and what we have in the stomach and so on. And so that's autonomic or involuntary. And then finally, under autonomic, we have two parts to that, which we'll cover when we get to the cranial nerves. We have sympathetic and parasympathetic. Most of you have probably heard of these before. We don't talk about them in great detail, unfortunately, because it's probably the most important part of the entire body, other than the skeletal area. That's what regulates our heart. It's what regulates our digestive tract and determines how fast we breathe. So typically, I'm going to put some generalizations here. What maintains homeostasis, which maintains our digestive system working and our heart at a slow rate, not a fast rate, is the parasympathetic. And so this is almost a cliche, is identified as the rest and digest system. So what you do normally, going to the bathroom, processing your food, the slower heart rate that we maintain is from the parasympathetic. And sympathetic is our stress. This is the danger one. All right, so when you're scared and your palms sweat, your heart rate goes up, and your skin goes pale, that's so you can run or fight. And so this is called the fight or flight system. So you're going down the freeway and you know you're speeding. The CHP car pulls up behind you and the lights go on. What happens to your heart rate? It goes up. And then you move over thinking they're pulling you over, and they just speed right on by because there's an accident that they're going to. Okay? So the norepinephrine has to be taken down, broken down and stuff, but your heart slows down faster than that would normally take. And that's because the parasympathetic system comes into play and releases neurotransmitters that slow your heart back down again. We're going to spend a little bit of time on this, mostly on the cranial nerves. We don't have any anatomy really that deals with the autonomic nervous system. Unfortunately, they don't make models of the autonomic nervous system. At AR, with the physiology involved, I've gone from spending 1 day talking about that to 4 days. Because when we go into our second semester, we talk about this with the control of every system. Whether it's talking about the reproductive system and erection and ejaculation, emission, to the heart, to everything is based on that. So but that's the physiology aspect of it. Okay, so this, you have a cleaner looking chart in the lecture notes, and I have the PowerPoints online. I just need to figure out how to make them. Can you guys see them? Because last semester they were out of, they're hidden, so I just need to go into, they're already out there, I just need to make them available. So I'll try to get that done today. Any questions on this? Okay, so for lecture concepts, if I say the femoral nerve, which as you recall innervates muscles of the anterior compartment of the thigh, is that a component of the central nervous system or the peripheral nervous system? It's going to be peripheral because it's a spinal nerve. All right. Is it sensory or motor? You're going to hear a lot of those types of concepts. This is a sensory. This is a motor function. Femoral nerve, sensory or motor? Obviously, we've talked about it for motor. But it also is going to contain within it the sensory fibers from the surface and the skin of the anterior thigh. So the femoral nerve would be both. All right. Trigeminal nerve. Is that sensory or motor or both? Primarily sensory, because we said it was sensory from the face and the nose and the chin and the teeth and the lining of the mouth. But remember, it was also motor to our chewing muscles. Okay. When we get to the cranial nerves, we'll be talking about the optic nerve, which carries sensory information from your retina back to your occipital lobe. Sensory or motor or both? Sensory. Okay. So when we put under visceral stimuli, we have a special senses that we include because we have organs that are involved with that. And so here we would have taste, vision or sight, hearing, balance, which uses proprioception and your inner ear. And I'm missing one. Smell. Thank you. All right, so we'll be referring back to this organization as we talk about different components, because that's something I emphasize a lot, is the function. Is it sensory or is it motor? All right? And you'll see that there's typically a pattern to our spinal cord and our brainstem. We'll see that today when I lecture about the spinal cord. Things that are on the ventral or anterior part of the spinal cord are typically motor. Things that are on the posterior or dorsal aspect of the spinal cord are typically sensory. We see that even when we get to the brain. The frontal lobe is where we have our voluntary motor. All the rest, temporal, parietal, occipital, are all sensory. Okay. So again, you might want to start making a list of keeping track or forming some way of of organizing that, because there will be questions, not just on the quiz on Monday, but throughout. Is this sensory or is it motor or both? Right? We work through its functions. Okay, so what I want to look at now are the cells of the nervous system. Obviously, we have the neurons. Remember, we're talking about tissue, and we kind of skipped the whole lecture on muscle tissue on our last unit, but muscle tissue is formed of muscle cells. We were talking about skeletal cells, but we have smooth cells and cardiac cells. Okay? Each organ, so the anterior belly of the digastric muscle is a muscle organ. Obviously, it's going to have connective tissue in the tendons and ligaments. It's going to have epithelial tissue lining the blood vessels. It has to have nerve to innervate those cells, and then the muscle itself. So we have all four tissues in that organ. When we get to the nervous system, it also is a nerve tissue. So after this unit, we will have covered all four tissues. Epithelial tissue, connective tissue. We won't have done blood yet, but we would have covered the concept of connective tissue, muscle, and this unit we're doing nerve. So when we look at the cells of neurons and their supportive neurons, that's what we're covering with the nerve tissue. The organs would be the brain, or the brain stem, or the spinal cord, or the nerves. And those would contain all four. The epithelial tissue and the muscle and the blood vessels. The connective tissue wrappings around the nerves have special connective tissue, and the nerves have wrappings around them as well externally. So that's our fourth tissue. And in addition to a typical neuron, which I'll diagram in a few minutes, we have supportive cells. May I raise the board? Bless you. So we have 5 types of supportive tissue. And these cells are called neuroglial cells. 4 of them are found only in the central nervous system, in the brain and spinal cord. And the 5th is found only in the peripheral nervous system, amongst our peripheral, our spinal and cranial nerves. What's the first one I have on here? All of these have the appearance of an octopus. All right, so they all look very similar. Their location is what you have to go by in determining, identifying them. So on the lecture exam, I'm going to pull up when I'm done talking about these, kind of a diagram of a slice through the central nervous system, brain or spinal cord. And we'll look at where we find these cells. So I will have a diagram, and I will ask you to either name the cell or identify its function. Okay? And so looking at its location, not just its appearance and size, will help you determine that. So astrocytes are not quite the largest, but the second largest of these cells. As you can see in its name, astro refers to star. And so these cells have very long processes. It looks stellate. These are not neurons, but a special type of nervous connective tissue known as neuroglia. When someone has a brain tumor, neurons do not typically divide, and so tumors are of these neuroglial cells. All right, they're equivalent of the fibroblast cells. And neuroglial cells are found kind of anchored onto blood vessels or capillaries. Capillaries in the brain have lots of cell junctions holding the cells together. And so they are considered to be tight junctions. There's very little that can leave a capillary. It has to be very small, which makes it difficult to treat problems in the brain with chemicals or antibiotics, because they are not able to leave the capillaries. And so the astrocytes help to maintain this blood-brain barrier. And And then they're also found associated with the neurons. So the neuron, or the nerve cell, not only do they maintain the environment by removing ions and used up neurotransmitters, but they will even recycle the neurotransmitters. So some neurotransmitters, these are the chemicals that neurons release to activate other neurons. If they stay out there in the extracellular fluid, they'll keep activating the neuron, so they have to be removed. And astrocytes help to do that. And they'll often recycle it and give the broken down recycled components back to the neuron so they can be used over again. Right? Microglial cells? Microglial cells look very similar to the astrocytes. They are these star-shaped cells, but they're much smaller. I'm going to draw that in a different color. Your nucleus doesn't look like an M. I'm just putting that there so you can identify it. Microglial cells are the phagocytes. They're the macrophages, if you will, of the connective tissue area of the nervous system. So these are phagocytic. They remove cellular debris. If there happen to be parasites in the nervous system or neurons that have died, they will endocytose it and break it down. Oligodendrocytes or ependymal cells are unique. They're only found in one location, and that is surrounding the fluid-filled space of the spinal cord. So in the spinal cord, we have a central canal, and in the brain, we have ventricles. And these contain cerebral spinal fluid. Not sure if that's showing up on camera very well. And so the ependymal cells either make this fluid or they help to maintain it. So they are found lining the space, and they're cuboidal cells that have this little tapered tip. So on. So they're going to take the fluid from the capillaries and modify it, prevent the blood cells from passing through, change the glucose and protein and sodium content. And so they synthesize and maintain cerebrospinal fluid from the blood. Now the fourth type of neuroglial cell, I'm going to draw over here because I need more room. This is called the oligodendrocyte. And so it has multiple arms, just like we've seen with our astrocyte and our microglial cell. And the oligodendrocyte functions to myelinate meaning it provides a lipid membrane covering wrapping around the neuron fibers. This plasma membrane of the oligodendrocyte contains a high number of phospholipids, as do all plasma membranes. But these lipids are known as sphingomyelin. And the cells wrap their processes tightly around the neurons, kind of squeezing out the cytoplasm. So all we really have is just layer after layer after layer of this lipid-rich sphingomyelin. This is in a physiology class, but what that does is prevents the ions from leaking out of the cell's, the neuron's membrane, and maintains a rapid speed of current transmission. An electrical current is how the nerve sends a signal. And the higher the difference in concentration, the faster that current flows, because that's what electricity is the flow of charged particles. And so if you have an electrical wire, and it's a bare wire, and it's touching a variety of things as it passes through the house, it's going to lose some of that strength as those electrical currents going to leak out to whatever it's touching, water or whatever. You wrap it with rubber or plastic, and it insulates that current. All right, and that's what the myelin sheath does to our neurons, is it insulates them, and thus it has a faster rate. So if we have a neuron, it has, we'll draw a typical neuron here in a moment. It has typically along one or more long processes, and the current is spread along this process. The oligodendrocyte will wrap around we'll send a process that will wrap multiple times around that axon and create a myelin sheath. It might send another process to another axon of a different neuron. And wrap around that one. So one oligodendrocyte will send out multiple processes that will wrap around multiple neurons. Okay. Unlike our peripheral nervous system myelinating cell, which the entire cell wraps around one fiber on a neuron. So this, I'll show you an image in just a moment. We have the same process occurring, but a different ratio. Okay. So let me pull the screen down and show this to you. So on the lecture exam, you will see an image similar to this. I don't think it's exactly like it, but similar to it. And I will have 2 or 3 of the cells marked and identify cell number 1 as, or the function of cell number 3 is, which are the following. Get a whole other lecture by the time I wait to explain it. All right, so up here is the central canal of the spinal cord. It's a small opening about the diameter of those wooden pointer sticks we have in lab, but it could also be a ventricle of fluid-filled space in the brain. The light blue cells are the neurons. They're large nucleus and nucleolus. And so if we look around, here would be a blood vessel. So here's an astrocyte with multiple processes extending between the nerve cell and the blood vessel. Those don't move around as much as the microglial cells do. So these can fit in between spaces and move around for their phagocytic activity. Here are the ependymal cells, and they're also associated with the capillaries because that's where they obtain the fluid that they're going to modify to become CSF. And then here are the oligodendrocytes. So the cell body sending out multiple processes, wrapping multiple times around the axon fiber, creating these layers of myelin. And we'll look at that with an individual neuron outside the central nervous system in a few moments. So those are the neuroglial cells. The difference between the oligodendrocyte and the Schwann cell that we see in a typical neuron found in the spinal nerve is that here we have the Schwann cell, the entire cell wrapping around the nerve fiber. So here's the nerve fiber, and the Schwann cell starts to wrap around it, and it continues to wrap around with the nucleus being external. And here on the electron micrograph, the black layers are just the lipid. It's been stained with a special lipid stain in multiple wrappings. So one entire Schwann cell or neurolimocyte is going to wrap completely around the fiber rather than having multiple wrappings. But the effect is still the same. You have layers of myelin, okay, and that increases the rate of conduction. Even neurons in the periphery that are not myelinated are still intimately associated with the Schwann cell. So you can see that these axons don't have the multiple wrappings, but they're still kind of surrounded, external to, but wrapped around by the plasma membrane. So they're not in contact with the cytoplasm, but wrapped around by the plasma membrane. Okay? And so what happens in the peripheral nervous system, if a nerve fiber is cut, the Schwann cells release growth factors that stimulate the nerve ending to grow back and also guide it as to where its end destination is supposed to be. Remember I talked about killing off the fibular nerve, and that dies back almost to the spinal cord and then grows back towards the muscles it needs to innervate. They've done experiments, and this is a long time ago, so they're still working on it, where they would take the chemicals, the nerve-like growth factors, from the Schwann cells and put them in a petri dish with spinal nerves from the spinal cord, cut off their axon, and while that wouldn't grow back in the spinal cord, in a petri dish with the chemicals from the Schwann cell, it does grow back. And so there's an environmental chemical difference in this location. All right, I'm going to spend a few minutes looking at the structure of a typical neuron, and then I want to do the cross-section of the spinal cord. There's a few things we'll have to cover in lab as far as the shapes of neurons, but that's relatively easy to do. So the typical neuron that I'm going to diagram, by its shape, is identified as a multipolar neuron, meaning it has multiple processes. In lab, we'll look at bipolar and pseudo-unipolar neurons, which have one or two processes. So we start with the cell body. And it has extensive branching for the purpose of increasing surface area, because it is through these components that a neuron is able to be activated, excited, or inhibited from creating an electrical current. So this is known as the cell body. It's going to contain the typical organelles that we see in a cell that is making proteins. So it's going to have a nucleus. And neurons have a very prominent nucleolus. You guys remember what's in the nucleolus? RNA. And the nucleus itself is the DNA. So that's the pattern for the proteins. What's a very characteristic and prominent protein that neurons need to make? They release it from the opposite end. This is being cut off about 20 feet. So they're going to release it from these swollen bulbs. Neurotransmitters. So the neuron has to be able to synthesize neurotransmitters, and it has the gene code for that. So when we make proteins that are synthesized from a cell, what are the two major organs that we need to have?
ER.
Ribosomal ER, because that's where the RNA is going to bind to and insert the new proteins. There are so many of these, they are so dense, that they're one of the first locations that ribosomes were identified, ribosomal ER was identified. And they're also known by the scientists that describe them as missile bodies. So these clumps are actually visible in the light microscope. And then, of course, processing them, we're going to have Golgi bodies. Present in the plasma membrane of these branches, which are known as dendrites. These are the receptive areas. The dendrites and the cell bodies have receptors that are capable of binding the neurotransmitters released by other neurons. So another neuron would send, let me do it in a different color here, would send its process to form a synapse and would release neurotransmitters that would bind to these receptors. And that would trigger a flow of current that may or may not be strong enough to trigger a flow through the rest of the cell. So what I want you to know, even though this is not a physiology class, is that the dendrites and the cell body together are the site where we find these receptors for neurotransmitters, and these are known as ligand, which means chemical gated channels. Okay.
Can you repeat that again? Sorry.
The dendrites and the surface of the cell body is where we have these receptors for the neurotransmitters. In other words, that receptor is going to be an ion channel that is closed until it receives a receptor binding to it. And then when that receptor binds to it, it opens. So it's a ligand or chemically gated channel. It requires that chemical to bind to it before it opens. Okay? If enough ions enter into the cell, they create a strong enough current that when it reaches this structure, it also has channels, but these channels will only open or close in relationship to the number of ions passing through it or the voltage. So it's kind of like a series of dominoes or video cassettes. You push one over, and that has the force strong enough to knock over the next one. So if the current reaching this area is strong enough to open this channel, it will allow enough ions in that will be strong enough to open the next channel, and so on. And our current will self-propagate all the way down the axon. So this structure is known as the axon. And it's the site of voltage or current gated channels. Okay. We have a special area right here where it tapers to meet the diameter of the axon. And this is identified as the axon hillock. It has voltage gated channels, but these have the lowest threshold. It takes the weakest current to open them. So it ensures that even if we don't have a really strong current, we can at least get those open, and then the rest of them will open. So this is our axon. It doesn't contain all these protein synthesizing organelles, but it will contain mitochondria because of the sodium-potassium pumps required to return the membrane back to its normal resting potential. We have a lot of microtubules because that's how sacs of neurotransmitters are transported, kind of like an L train. They're kind of moved down the axon. They get to the end of the axon, and these branches, instead of being known as dendrites, are known as axon terminals. Or you might see the word telodendria, or a bunch of them. And each dilated structure contains those sacs of neurotransmitters and is known as a synaptic gluton or involve. And that's what I drew over here. So when we see a cell body in our models, we'll also see those glutons from other neurons synapsing with it. All right, so if this was a motor neuron, which multicolors are, its cell body would be right here in the spinal cord or in the brainstem, and its axon that I've drawn would go out of the spinal cord to whatever gland or muscle it was going to innervate. Innervate means provide nerve function too. So some of the early axons that were studied came from giant squids, and those cells are over 20 feet long. So the neuron that you have in your lowest portion of your spinal cord, sacral spinal cord, will send nerves to the muscles on the bottom of your feet to flex your toes. That's how long that nerve cell is. Okay? 3 feet, 4 feet, 2 feet, depending how tall you are. So that one cell is going to travel that entire distance. All right, so in lab we have 2 or 3 different types of models of the neuron and these structures that you can find on them. We'll also talk about the different shapes of neurons. So this is a multipolar. We'll look at illustrations of additional ones. In fact, let me just do that really briefly, because I make a change in what is in your lab manual. So when we look at types of neurons, we just drew the multipolar, which has dendrites and a long axon. Your lab manual uses the term unipolar. A true unipolar neuron has no dendrites and one process sticking out. That's not the unipolar that your lab manual is talking about. I'll get to it, okay? But that's the true unipolar. There are very few unipolar neurons in our body. Then we have bipolar. We're not talking about a psychiatric order. These are sensory neurons. The information would travel towards the cell, around through the cell, and then away from the cell. And these are sensory neurons. 2 areas in the body where we find them: in the retina of the eye and the olfactory neurons in the nose. They're called olfactory epithelia, even though it's really neurons. Now, watch carefully my fingers. If I take these 2 processes and I migrate them around and fuse them together, they form a single stop. Okay? But that wasn't their original origin. And so the next neuron looks like this. The one I've been drawing on the board. This is what your lab manual is referring to as a unipolar neuron. No, it's not actually. It is a pseudo motor neuron. And except for our bipolar neurons, this is all the sensory neurons. Will be pseudo-unipolar. We have several charts in lab that will illustrate this.
Question.
Yes.
What do you find is the unipolar?
Unipolar are found in the cerebellum,
Okay.
certain isolated areas of the brain, but not in the peripheral nervous system. Alrighty.
So all of the three are found in the peripheral?
Yeah, these would be forming when we talk about the spinal cord. I'll locate these for you. Okay. In the central nervous system, we find a lot that are multiple. They look like this without a long axon. Okay, so we're talking mostly the peripheral neuron. May I erase this diagram of this neuron itself? Okay, the location and parts of the spinal cord I'll go over in lab, but it's specifically the cross-section of the spinal cord that I want to cover. The spinal cord begins at the level of the foramen magnum. It does not extend all the way down to the sacrum, but it extends to the level between the first and second bodies of the lumbar vertebrae, L1 and L2. The spinal nerves have to go farther, all right? But we'll cover that in lab. So cross-section through the spinal cord. Sure, I've got the right black for this. So this is ventral or anterior. And in the nervous system, we go back and forth. So we'll talk about the ventral ramus, but the anterior gray horn. Just the way it is. And I will accept either term. Okay? So if you want to call it the anterior root and the ventral gray horn, it's not common usage, but they essentially are the same as far as the nervous system is concerned. So ventral anterior, that's going to be mostly motor function, and dorsal or posterior. The spinal cord is made up of a bunch of cell bodies which are never myelinated. So structures that are not myelinated, if you were to cut a spinal cord and look at it, are kind of a grayish brown color. So they're called gray matter. And the myelinated fibers look white. So the gray, the gray matter is found in H form. Again, these are cell bodies that we're looking at in cross-section, but this would be a structure that would run the entire length of the spinal cord. And we're looking at the thoracic level, or our typical spinal cord. So everything within that are unmyelinated cell bodies. There might be a few strands of myelinated neurons, but they're not enough to show up as white matter. Okay. So I'm going to label this side of it, and then when I'm done going through the labeling, I want to put the neurons in to help explain the function. So this is in the back. And it's gray, so it's identified as the posterior. And I never can remember. Do you guys spell gray with an A or an E? I always do it the opposite. An A? Okay. Posterior gray horn. It's made up of the cell bodies of sensory neurons, as you'll see when we come back to it. Okay? This is the lateral gray horn. And it's made up of autonomic, sympathetic usually, cell bodies. So it is motor. And then these large ones in the front are where we find our skeletal muscle neurons. So this is known as the anterior gray horn, and this is voluntary motor. So the cell bodies going to your intercostal muscles, and their intercostal muscles, or going to the flexor carpi radialis, are going to be located right here. Have you ever heard of polio? Polio is a virus which destroys these anterior cell multipolar neurons. Therefore, descending information from your brain will get there, but it won't get out to your muscles. Okay? It's going to be on this side too, but I'm going to use this side to diagram the nerve pathways. This band of gray matter in the middle of our H is known as the gray bridge, which is referred to as the gray commissure. Commissure are bridging components. And then this opening here is the central canal. And that would be lined with ependymal cells that we looked at earlier. All right, the white matter surrounding it is ascending and descending nerve fibers, most of which are myelinated. And so these are identified as columns. Even though we're looking at it in cross-section, everything is going up and down. Unlike in many of our nerves, which are mixed, you cut one nerve, you're going to have a spinal and motor components that are lost. While we might find sensory and motor components here, they're distinct. So we don't have a mixture of sensory and motor tracts. We have a sensory tract and a motor tract. They might be side by side, but they don't mix. This area of white matter back here is identified as the posterior, or dorsal is a common term, posterior white column. Since it's posterior, it's sensory. It has a unique function. It's completely sensory. Don't have any motor tracts there at all. And this is sensory for pressure, non-distinctive touch, and for balance. There is a virus, a rare virus, that can destroy that area as well. And these individuals don't have any joint sense. When their eyes are closed, they have no idea whether their fingers are flexed or their knees are flexed or extended. So you can imagine how hard it would be to walk and do things unless you were actually looking to see what was going on. Six is the lateral white column. And this is both motor and sensory. Again, distinct sensory tracts and motor tracts. So you would have a motor tract, a sensory tract right here, a motor tract right here. But you have distinct motor and distinct sensory tracts in that area. And then 7 is known as the anterior white column, and that is ossivote. In this area through here, we have white fibers crossing over, and this is known as the anterior white commissure. It's going to be primarily sensory, but it has some motor. So it's usually sensory fibers that we see crossing over there. These are known as the anterior and posterior median sulci, or fissures. I don't name them because they go opposite their definition. Usually a sulcus is shallow and a fissure is deep, and those are reversed here. But anterior median, posterior median. In the front, in the middle, in the back, in the middle. It's pretty obvious. All right, now let's identify the structures that are just outside the spinal cord. We have this structure right here is the spinal nerve. It receives or gives to the spinal cord 2 roots. So in the front, this is identified as the ventral root, and in the back, the dorsal root. The ventral root is motor only. The dorsal root is sensory only. Spinal nerve is both. And then the spinal nerve divides into 2 arms or branches. And our word for arm or branch is a ramus. Remember that term? So this would be the dorsal ramus. Remember we talked about nerves to the splenius capitis or the erector spinae were the dorsal rami? That's what we were talking about. Okay? And then this would be the ventral ramus. And this is both. Sensory and motor. And the ventral ramus is also both. Okay, so I want to illustrate this with nerve fibers coming in. We'll do the sensory first because that's what our body responds to with movement. So sensory can come in either the dorsal rami or the ventral rami. Because remember, they're mixed. So if we have sensory coming in from the middle of our back, there's about 4 inches on either side of your spinal nerve column that's going to be innervated for pain, touch, and temperature by sensory neurons. So those are going to come in from the back. And remember, they're pseudo-unipolar neuron. So their cell body is located in this swelling. And then the rest of the neuron is going to come into the posterior gray horn. So this swelling is created by cell bodies outside the central nervous system. And as such, it is known as a ganglion. The ganglion of the dorsal root. So it's known as dorsal root, not sure I very well, ganglion, and that is sensory only. neurons coming in from, say, my anterior abdominal wall or from my hand, depending on what level, coming into ventral ramus. So dorsal ramus is only going to be for that strip of skin and the deep muscles below it on either side of the spinal cord. Everything else, front of the trunk, side of the trunk, limbs, is going to come in the ventral ramus. It's sensory. So even though it comes in the ventral ramus, passes through the spinal nerve right here. I drew it a little bit shorter. Passes through the spinal nerve. It's going to go into the posterior dorsal root. And so we're going to go into the posterior gray horn. Right? Now it can synapse there. Neurons in that that's what make it gray, are the cell bodies that those neurons are synapsing with. And these neurons will cross through the anterior white commissure to the other side of the spinal cord and will go up to the brain. So a whole bunch of these neurons doing that create a sensory tract. That's why the lateral white column is white. Okay? Now obviously neurons coming in this direction are going to cross over to the other side. I'm not drawing that in because it gets even messier. Okay? Now some of these neurons coming in, if they're coming in, say, from my elbow and giving a joint information, will not stop. They will just go up the posterior white column. And that's why the posterior white column is all sensory. Now let's draw motor. Motor neurons cross over, 85% of them cross over in the brainstem, and then descend on the same side of the spinal cord. So we're going to have motor neurons coming down the spinal cord. Their cell bodies would be in my frontal lobe of my brain. Okay, we'll review this when we get to the brain, and we'll keep coming back to this pathway so that makes sense. So we're going to have a motor tract. We would have a similar motor tract over here, distinct from our sensory tract. These neurons are going to go into the anterior gray horn and synapse with the neurons in the anterior gray horn. Where do they go? Out the ventral root. If they are going to innervate the erector spinae muscles or the splenius capitis, they would go out the dorsal rami. If they are going to go to muscles of my chest or my arm, depending on what level I'm at, they will go out the ventral rami. So now when we go back and look at our terminologies, you can see that the posterior gray horn has sensory cell bodies. That's why it's sensory. The lateral gray horn also has sensory gray bodies. An anterior gray horn, motor cell bodies, and anterior gray horn has motor cell bodies. So lateral and anterior gray horns are motor. When we look at the white matter, we have sensory neurons traveling only sensory neurons traveling in our posterior white column. Both motor and sensory traveling in our lateral white column. And I did draw them crossing over, but both would be here as well. And you can see that the dorsal root and the ventral root are separated by function. Dorsal root is only sensory. Ventral root is only motor. But in the spinal nerve, they are mixed. And in the branches of the spinal nerve, they are mixed. So on the quiz on Monday, you will have a diagram that looks similar to this. I will give you copies in lab. And I will have various structures numbered. So if I have this number 1, identify this structure, you would say spinal nerve. If number 2 is this structure, and I say is it motor or sensory, the answer would be both. Okay? Those would be the 2 types of questions that you would see from this diagram. All right. I'll see you in lab.